Okay, so in the last class, I gave you a description without getting into the molecular detail of how gene knockout can be done in mouse, right? And we were very categorical that we are talking about gene knockout or gene knock-in and not transgenic, okay? So continue on it. So if you can, so like this technology after it was developed, it actually opened up so many research avenues. Okay, the first thing is that this development of this technology was almost congruous with development of the ES cell technology and this entire field of regenerative medicine. Like you know that ES cells can be used to develop any kinds of cell or tissue type. There have been research laboratories who dedicated their entire careers just to specify the culture conditions by which a ES cell can be driven to muscle fate or bone fate or neuron fate or pancreatic lineage fates and stuff like that. So that is one area that definitely is an outcome of the development of knockout mouse technology. I'm not saying that for knockout mouse technology per se, but that the fact that ES cell could be isolated and cultured and that was used in this context eventually spun off into an independent discipline. Okay, and then of course functional genetics. Earlier, all one could do is to study known, like spontaneously occurring mouse mutants, and to figure out, what may have, which gene may have been mutated and why that gene's loss of function or gain of function caused certain phenotypic changes. Those are the kind of questions that scientists could ask. Now suddenly, scientists are empowered and they can ask questions just like they could ask in the East. That is, what is the function of this gene? If I knock it out, then how will the physiology of the animal change? That will give them some idea about what is the normal function of the gene is. So in this context, I will show you some of my own data, just because it was much easier for me to access and explain to you. So I was interested in one such question. Okay, so that question was that to give you a little bit of history, a long time back in 1960, there was this scientist called Marshall Urist. He was a physician scientist. He was trying to develop, biomaterial that could act as vehicles that will release drugs very slowly. So slow release drug vehicles now are very popular and common. You may know about birth control pills, which can be injected only once in a year, and that will provide protection for a whole year, or certain steroids or painkillers, and many kinds of cases like insulin. There can be slow release versions of insulin. But we are talking about 1960s when such material development, such genre of research were not really done. So Marshall Urist thought that bone has a very nice woven structure. Okay, so he thought that why don't I why don't I use the bone after you have removed all the biological material from it as a slow release vehicle. So for that purpose, what he did is For that purpose, what he did was that he took bone from, I think, a rat or rabbit and demineralized it, made a pulp of it, and then, as the first experiment, put some placebo in it, and just to see, it will not have any effect. And to his horror and surprise, he saw that wherever he implanted that demineralized bone, the entire surrounding area became ossified. Like it was put in muscle, the whole thing became bone. So that gave him an idea that there is some kind of an biological activity in the demineralized bone that induces new bone formation. Okay, so then, to cut a long story short, that biological material was eventually purified biochemically, characterized, sequenced. The protein was sequenced from the gene sequence was learned, and it turned out it was called, that activity was called bone morphogenetic protein, or BMP. And the first when it was discovered, it was just called BMP. And then when the DNA and protein sequence got to be known, then they realized that this is very similar to a well-studied popular gene in Drosophila called decapentaplegic, or DPP. Now, DPP is a very, very critical developmentally regulated gene, and it is important for patterning of Drosophila embryo. So, so it was learned that BMP is basically a vertebrate homologue of Drosophila. Then within few years, when other scientists followed on this work, eventually genome was sequenced. Now we know that there are 20 different BMPs in vertebrates. Okay, like in humans, we have 20 different BMPs. Drosophila has just one BMP, one DPP. And we have 20 different BMPs. Now, even if you take recombinant BMP protein, now you know how to make recombinant protein. If you take recombinant BMP protein and put it under the skin, like in the area called dermis, it can induce ectopic osteogenesis. So it was kind of expected that BMP signaling is sufficient for bone formation. The question is it necessary? So the moment you ask the necessity question, what you have to do is that if you don't have BMP, do you still form bone or not? So that was the question that was dogging scientists for a long time. The problem was that there are 20 different BMPs. In the limb only, in the hands and legs, in the limb only, there are 5 different BMPs that are expressed. Now, when they were knocked out individually, there was absolutely no phenotype. And if you and if there was a phenotype, the embryos will be dead very early on, because they figured out that BMP signaling is important for heart development as well. So heart develops much before anything else. So if the heart development is not right, mouse is dead. If it is a dead mouse, you cannot ask the question whether it is necessary for bone formation or not. And then you have the problem of redundancy, that if you knock out one, the other BMP homologues might just step in and do its job. So I will tell you how it was done more in molecular genetic detail. But just to cut a long story short, what I could do is that I knocked out 2 different BMP ligands just in the limb bud. If you do just in the limb bud, then heart development will not be affected, or any other essential organ development will not be affected. So the animal will survive. And because you were knocking out 2 different ligands, we got lucky. So we got severe phenotype. Okay? You can see that this mouse does not even have a properly formed hand, and just the foot can be seen, not really the whole leg. Okay, so this was a demonstration that BMP signaling is necessary for bone formation as well. Okay, so then what happened So this is how you can do it. Now, along the same line, so this is how you can ask questions, functional genetics questions. Also, either fortuitously, that is, you knocked out a gene and you saw there is a lot of cancer, so you know that this is a good model for cancer, or you know that in cancer this gene is mutated, like from human tissue analysis or something like that, then you knock it out or impose exactly that kind of mutation that is seen in the human cells, and then you reproduce cancer in mouse. So that becomes a very nice cancer model. You can, study efficacy of drugs, efficacy of radiotherapy, chemotherapy, all sorts of things you can study. So therefore, mouse knock-in technologies opened up the possibility of disease models, cancer and other disease models, cystic fibrosis. Even there is OCD model for mouse. Like there, what they call it is that obsessive compulsive disorder for grooming behavior, meaning they keep on scratching their hair to make it nicely flat on their body surface, and that's the grooming behavior, and they do it so much that they end up losing all the hair just because of the scratching. So all sorts of disease models are made. In my work, fortuitously, we created a model for osteoporosis. So I told you a little earlier that we knocked out 2 genes simultaneously and we got no limb, right? But if we knocked out only one ligand, okay, then the mice were born looking quite normal. Like you cannot obviously tell any difference. And then, to put it in the context of an interesting story, one day the mouse technician in the US, in the mouse facility, there will be a technician who will feed the mice, give the water, change the cages and stuff like that. The mouse technician came to me and said, Amitabh, your mice, they're acting funny. They do gymnastics. I go, what do you mean? So I went down with her and I realized that the mice have really strange walking pattern. So then we ran some X-ray on these animals. And as you can see, that depending on the age of the animal, you can see some very clear fractures. Okay? And you imagine that these mice did not have any serious fall, nobody broke their bone, nothing. These were happening simply by walking in the cage. So this is what is referred to as low impact fracture. Low impact fracture is a hallmark of osteoporosis. Okay? Osteoporosis is a disease particularly prone in postmenopausal women, and there they lose their bone mass. As a result, they develop low impact fracture. Like if you just put the hand on a table a little bit hard and you can break the bone. Okay, so then we studied a lot, we induced fracture, and we realized that the mutant mice cannot do any fracture healing at all, while the wild type, even if you have lost one copy of the gene, even then there is fracture healing response. But if you lost both the copies of the gene, there is no fracture. So now we have used this, and we have proposed some osteoporosis therapy theory for it, of course, and stuff like that. So this is how knockout technology can help you a lot. Okay. So now, what you should know is that just like you can take out a gene, you can insert a gene as well, knock-in technology. Okay. So by that, so when you are knocking in, that there also you are knocking in at a particular locus. You know exactly in which chromosome, at what location you are putting it in. That's called knock-in mouse technology. And this is an example where a gene called double curtin was knocked in. and the size of the brain grew dramatically. Of course, scientists occasionally do make some catchy cartoons and headlines. So there's that it's a super mouse, with a big brain and etc. So, but the fact is that you can study gain of function as well using technology. Like, it is actually knockout technology, but just because it is a gain of function, it is often called knock-in technology. So all these slides that I showed you, this was in the context of presentation to the entire faculty of IIT Kanpur right after mouse genetics work earned the Nobel Prize. So I was requested to make a presentation to the whole faculty to explain what was the Nobel meaning work. And in those days, there was very, very good PhD student in Professor Katti's lab, Rajesh Bashita. He helped me in decorating the slides, and this is a fantastic cartoon he made, and I still treasure it. Rajesh is now a faculty in Central University of Gujarat. Anyway, so now let us get into some of the technical nitty-gritties, that how a gene is targeted. So let's first do it with yeast. It is very easy for you to understand. And it is, just notice this, that only 20 nucleotide flanking sequence, that's all that is needed. So this is the flanking sequence that will dictate homologous recombination. Okay, and in between what you have is the target gene. Okay, so you can simply take primer for kanamycin resistance and use this flanking sequence, and then similarly you have another primer that will also be overlapping with kanamycin resistance cassette and the flanking sequence. So you can design little long primers and then do a PCR, and the PCR product is kanamycin resistance in the middle and the flanking sequence here and here. Okay, these flanking sequence are exactly the same sequence as the location in the genome where you want the kanamycin cassette to go in. Okay, so you simply insert that piece of DNA in the diploid cells using transfection technology, and then you select on G418. If the yeast cell has integrated kanamycin resistance cassette in its genome, then it will be able to form a colony in there, right? So you select that, and then when you sporulate, then the two kanamycin resistant, there will be two haploids where the gene is knocked out and two haploids where the gene is not knocked out. So just tying back to the course that we have discussed so far. So the ultimate proof that a particular gene is involved in a particular pathway. So you will do is genetics, you will do random mutagenesis screen. After that, you will rescue it with the library plasmid, and eventually you will see that that's how you will identify the gene that was mutated towards a particular assay, right? But there is always a possibility that is there that the actual mutation was somewhere else. So the final proof, which we call the acid test or golden test, is that gold standard test is that you actually, now that you know the gene sequence, you simply knock it out in a wild type cell. And if it reproduces the same phenotype as the original mutant, then you are confident that the gene has one and only one function, and that is the function for which you identified it in the first place. So that's how you can do. You can also use this kind of technology. Just imagine this kanamycin cassette is a little longer. There is also GFP right here. So you can make these cells that will fluoresce, okay? The GFP positive cells. Or you can induce any kind of functionality. So this is how you can do knockout, knock-in in these cells. The mouse one, philosophically, it is not very different. It is the same thing. just because the homologous recombination efficiency in mouse is much, much lower. So therefore, things get a little complicated there. How? One is that the sequence, that homologous sequence, which is only 20 nucleotides in yeast nowadays, often runs into 3 to 5 kilobases in mouse. Okay, so you are talking about really complicated cloning. And then this is the homologous sequence, this is the homologous sequence, and within which you have the neomycin resistance cassette. It will be important later on, just to let you know, that this neomycin resistance cassette will have promoter enhancer of a gene called phosphoglycerokinase. So it is basically called PGK neocassette, phosphoglycerokinase neomycin resistance cassette. So that is the so that this cassette is self-sufficient in terms of expression of the neomycin resistance gene. And then please note, on the other side of the homologous region, like selection cassette, homology arm, then there is another cassette inserted there, which is called HSV-TK. Okay, so this is actually a gene called thymidine kinase. It is its expression. I think it is isolated from herpes simplex virus, and this gene, if it is expressed, then those cells where this gene is being expressed, thymidine kinase, they will not be able to grow if there is a chemical called jancyclovir that is present in the solution. Okay, and you will see the importance of that. So, as I said, that the homologous recombination efficiency is fairly low. So you need a long homology arm cassette. And if there is a good homologous recombination that you can see here, so there is this, the region where the recombination is taking place. So you can see that the neomycin resistance cassette is inserted there. And because the crossing over happened within the homology arm, therefore the yellow part, that HSV-TK part, is no longer, it could not become part of the chromosome. Okay? So it was hanging out, so it is lost. Okay, but if there is, so the way it works is that you transfect the cell with the knockout construct, and then like it has shown schematically, there are three populations. One, non-recombinant cells, like either the transfection did not happen, or even if it happened, there is no recombination, the DNA fragment was chewed off. It is not a plasmid, right? It's a linear plasmid, chewed off or whatever. In some where the recombination happened exactly as it is shown here. And in some, recombination happened, but not exactly as per the homology arm, but in some random region, okay? Some random region. As a result, so now it is random, right? So it is not limited to within this length. So this HSV-TK part can also become part of the chromosome. If that happens, like depicted as these cells, so when you first grow the cells in presence of G418, because of the neomycin resistance cassette, all the white cells will be eliminated. Only the green and these cells will be there because they have the neomycin resistance cassette as well. Then you grow them in presence of jancyclovir. It is called a negative selection, that where the recombination did not happen the right way, there the HSV-TK cassette will also be part of the chromosome, and as a result, these cells will be eliminated and only these cells will prevail, and that's your targeted cell. Okay, so once you have isolated the targeted cell, then you go for blastocyst injection. As I described you earlier, here where it is shown is that these are the black mice. So there the gene that you are trying to target is say X. Okay, and this is the coat color gene. Okay, in black it is small a, in brown it is capital A, capital A. Okay, diploid, of course. Right? These are stem cells, and this is the blastocyst cells. Both are diploid, and this is their genetic nature. Now the modified ES cell, you have knocked out only one locus, right? Like you can imagine that you are selecting with, like like what is the probability that the transfection will happen and it will recombine in the right locus? Maybe 1 in 10,000, let's say, because you are doing a selection, so you can easily get it. Now, what will be the probability that both the copies of the chromosome will get modified the same way? That is 1 in 10,000 by 1 in 10, multiplied by 1 in 10,000. So that makes it very, very rare. So there are ways of doing that, homologous recombination, homozygous homologous recombination in cell culture. But generally speaking, is, at the end of the targeting, it's a heterozygous cell. So X minus X. So like I'm talking about this genotype, okay, X minus X. And in the black recipient mouse, it is X over X. Now you put those chimeric blastocysts in a pseudo-pregnant mice, pseudo-pregnant female. Please understand that we are talking about implantation of embryo. Any female mouse is not going to be able to, like is not going to, will not have the ability to implant the embryo. So you have to prepare the female reproductive system artificially by injecting hormones so that they are recipient and they can implant the injected embryo. So that's why they are called pseudo-pregnant female, meaning they are injected with hormones that simulate pregnancy-like situation in the fallopian tube. Okay. Or fallopian tube equivalent in mouse. Like I am really not fully confident about the physiology of that part. So that is they're transferred. Then this is called the foster mother. By the way, this is exactly the same technology that is used for in vitro fertilization in humans. Of course, you do not do this chimeric injection, but like the egg and the sperm are collected separately in a petri dish. There you do the fertilization, and then it is transferred onto the same mother, like who donated the egg in the first place. The mother's body will be prepared with hormone injection, so the pseudo pregnancy, and then it will be injected for implantation. Or nowadays you also know that there are surrogacy cases, like another lady will act as the recipient, but the same technology. Now, as we discussed in the last lecture, that once you've done that, then you can get one of the two kinds of pups. One, pure black pups, meaning the ES cells did not contribute to those embryos at all, most likely, or ES cells that have contributed to the embryos. That's why you get the chimeric ones. Now you take the chimeric mouse and then cross it with the wild type mouse. The chimeric mouse will have, like, think of the possibilities of the germ cells. Remember that these stem cells, these are X minus, X plus, and they are A over A, right? So the germ cell in the chimeric mouse can be A over X plus, like this copy, or A over X minus, or like if it is not from this stem cells, then the natural one, there it is only one possibility, A over X plus, because like the black mouse derived stem cells, like the natural blastocyst cells, there is no knockout, right? So it will be always small A over X plus. So these are the three kinds of germ cells that are possible. But if this germ cell or this germ cell, the one that produced the sperm, that's the only thing you can track. You cannot track anything else. And you cross it with the black mouse, brother-sister mating, then you will get either one of these two genotypes. It has to be capital A over small a, because the other mouse is small a. And it has to be either X plus over X plus or X minus over X plus. Okay, so you will get this. So now you know that the mutation has gone germline, and then you do RT-PCR exactly to know the genomic locus, like which we call genotyping, and then you cross them several times. Eventually, you will get a homozygous mutant mouse. If the homozygous mutant mouse is dead, embryonic lethal, then you cannot propagate it, right? In that case, you always have this heterozygous mouse in your colony anyway, so you can always go back to those. You may not be able to study a whole lot like what happened with the BMP knockout mutants. You could not study bone, but you got to know that this is an embryonic lethal mouse. You study very early stage events. Okay? So there are many ways you can do it. So this is how it is done. There is another way of doing knockout. I will not talk about it today. Okay, we are getting long. So you do up to this part from in this lecture. Then in the next lecture, I will tell you another variation of doing knockout. Okay, and knocking. Okay, okay. Bye bye.